Hi, everyone. My name is Andrew Bremer, and I'm excited to welcome you to the National Academies of Sciences, Engineering, and Medicine, and to our webinar. The National Academies provide independent, objective advice to inform policy with evidence and to spark progress and innovation. Our programs convene experts to share the latest research, to explore new ideas and possible solutions to confront challenging issues facing society. Aligned to that mission, we hosted a workshop in October on successes and challenges in biomanufacturing, sponsored by Schmidt Futures. You can find out more information about the workshop and watch the recording at the hyperlink above our live stream today. A proceedings detailing the workshop's discussions is also in the works, slated to be published in early 2023. We are eager to continue advancing the conversations and actions around key issues in biomanufacturing that were discussed during the workshop. Today's webinar on bioindustrial manufacturing scale-up capacity is the first of several follow-on events we'll be holding over the coming months. If you have any feedback, inputs on topics for future events, or would like to stay informed and aren't yet on our mailing list, please reach out. My email is located at the bottom of the live stream today. For today, if you'd like to ask any questions, please submit them anytime in the Slido box below the video, as we'll have a Q&A later on. With that, I am pleased to be joined by our moderator for today, Jim Philp. Jim is a policy analyst at the Organization for Economic Cooperation and Development, specializing in industrial biotechnology, synthetic biology, and sustainability. It was a pleasure to work with him on the workshop planning committee, and I am pleased to welcome him here now. Thank you all again for tuning in, and Jim, over to you.
Thank you, Andrew, and thank you also for the invite for the workshop in October. It was very enlightening and informative in an area where we need enlightenment and information. As Andrew mentioned, folks, today the webinar will focus on bioindustrial manufacturing scale-up capacity, probably the greatest enigma of all the aspects of policy that we face in this business is how to scale this up in the face of a fossil industry which is over 100 years old. The need for expanded scale-up capacity was discussed by several of the experts during that workshop in October. This webinar is also taking place against a backdrop of notable recent developments worldwide that have highlighted this issue, including the US Executive Order on Biotechnology and Biomanufacturing, signed by President Biden on September the 12th. A recent report from President Biden's Council of Advisors in Science and Technology, CAST, and further international activities like COP27 in Egypt, including the discussions on the bioeconomy as a climate solution, and that doesn't happen very often, doesn't happen often enough, with scale-up as a critical issue to be addressed. Needless to say, this topic is one meriting additional focus. As a side note, I'll mention that while today's webinar is focused on bioindustrial manufacturing, discussions during that workshop in October made it clear that the topic of today's webinar is also just as relevant and just as critical differences there, particularly about scale and about added value and margins, but the same issues pertain. But with that, let's get started. We've got 4 panelists today. After I provide an introduction here, we'll hear some brief comments from each of them, and then we'll transition to a discussion and Q&A with all our panelists. So each of the panelists has about 7 minutes, and I should get started straight away then. The first of our speakers is Deepti Tanjore. Deepti is the Director of the Advanced Biofuels and Bioproducts Process Development Unit, ABPDU, where she engages with scientists across industry, academia, startups to resolve scale-up challenges for synthetic biology-based technologies. Her research focuses on modeling the impact of bioprocess conditions on microbial heterogeneity and developing inline analytical tools for real-time adaptation process development in bioreactors. She holds a PhD in biological engineering from Penn State University. Deepti, the floor is yours for your 7-minute introduction. Thank you.
Thank you very much, Jim. And also thank you, National Academies, for holding this webinar on a very important topic. So let me share my slides here, and please confirm that you see what you're supposed to see.
Yeah,
yeah.
I see it. Yeah.
All right. So I'm as Jim mentioned, I'm the director of ABPDU. I have been at the ABPDU for about 11 years. I've seen quite a bit of the ramp up in the bioeconomy, biomanufacturing side of things. And today I'll give you an overview of what ABPDU does, and hopefully we can discuss more about the issues in this space. Excuse me. So at the ABPDU, we understand and solve scale-up challenges. We are supported by the Department of Energy Bioenergy Technologies Office, specifically to solve problems for our collaborators. Most of our collaborators are startup companies, but we work with larger companies and also academic entities. So the reason we exist is scale-up or conditions in bioreactors substantially different than those that are used at the lab scale. So if capillary effects in microtiter plates really inhibit our ability to understand how a culture experiences real process conditions during scale-up. So the smallest bioreactor we have used that can really give us an idea of what happens at large scale is at 250 ml, which is the amber bioreactor. And for fungal fermentations and waste feedstocks, we really have to use at least 2-liter scale bioreactors. So we have done this type of scale-up work for over 75 companies. We started in the biofuel space, as our name suggests, and we've helped companies then later on in materials and chemicals and food health applications. Especially during COVID times, health became a very high priority for us. So really our goal with all of these companies was to take their milliliters or maybe up to a liter scale technology and scale it up to hundreds of liters. So we like to say that we meet our collaborators where they are. Maybe they come in with technology at milliliter scale. We scale it up. We do a technology transfer and then scale it up all the way to 300 liters, which is our largest capacity. Or sometimes we work with companies that have a complete process development team and bench scale fermenters. So then they just come to us for the 300-liter scale. So we're very modular in that sense. The 300-liter capacity not only helps us understand what happens in a bioreactor, but also gives us enough volume to play with downstream processing. At the ABPDU, we have about a dozen downstream options in various categories, from centrifugation all the way to evaporation and drying. You see some of these pieces of equipment listed here in gray font and italicized because those pieces of equipment we haven't actually, we don't own them. We rent some of these pieces of equipment. So when we don't have a downstream unit operation, we work with the vendors, rent a piece, bring that skid on site, connect it to our fermenters, and help understand how scale-up separation will work for our companies that we're working with. So this is, I want to make sure that this part is clear to everyone. It's very difficult to decide what downstream unit operation you want early on. In fact, you have to really try a few different unit operations in series. Sometimes you have to, one single unit operation may not be enough, so you have to try a few. So that's why we have a dozen plus, and still we don't have enough. We still have to rent for our companies. And then when it comes to analytical equipment, we have some basic analytical equipment that we listed here, but we are also part of the Lawrence Berkeley National Lab, which really gives us the opportunity to go access these advanced analytical tools that are out there that currently are not being applied to bioprocessing. An example that I can give you is for a certain company, we used the Zeta Sizer at a molecular foundry to find out why we were not seeing protein on the other side when we were filtering it. So equipment like that, when we don't have it on site, we can access it at LBNL, and also the experts who actually know how to use the equipment and analyze the data that comes out of that equipment. So that being a part of LBNL gives us that substantial advantage. So as an FFRDC, we are federally funded facility. We really look to push the envelope as much as we can in terms of availability of equipment, novel new technologies that the industry needs but cannot access yet. So one such a piece of equipment that we've recently purchased, installed, and commissioned is gas fermentation capabilities. These are 8 500 ml bioreactors, HEL bioreactors, that we purchased, and then we had to do substantial customization on this equipment to be able to apply, to be able to use it for a variety of gas fermentations, everything from carbon monoxide, carbon dioxide, hydrogen, etc. These reactors also actually double up as pressure vessels for us to be able to simulate what happens in even liquid fermentations at very large scale. So at tens and hundreds of thousands liter scale, the lower layers of the bioreactors experience substantial pressure, and your culture behaves completely differently than it would at even hundreds of liter scale. So we need to test those conditions. And if we could do that without actually having to run a 100,000 liter reactor, that would save a lot of resources for companies. So I'm very happy to announce that we've actually used this equipment to do such studies for both liquid fermentations under pressure, and recently ran a gas fermentation for a company recently. So we're ready to use this equipment for industry. We have a bunch of companies lined up to use this capability at the ABPDU now. And another capability that is in progress in development is flow cytometry. What we are trying to do is we're trying to understand how microbial cultures evolve in a bioreactor over a period of time, and their evolution changes due to the different process conditions that we apply to them. So here you're seeing a rhodosporidium on day 1, and it's nice and budding, and then on day 5 it's really battered, and it's the culture is behaved the way it's supposed to, and it's now time to take it offline. But if we had an opportunity to understand how the culture behaves, can we control the conditions to prolong these biocatalysis so we can actually extend the process and derive the productivity that we want out of the culture? That would be fantastic. So to be able to do that, we are looking at imaging, flow cytometry, and a bunch of other tools that we can apply with our bioreactors while we run the fermentation as we currently do. So this capability, we're hoping to have this online for our industry partners by mid next year. So finally, I want to talk about a couple of industry projects that we have been successful at, and you can see a lot more information on this in the articles that I listed here. I'm happy to share these articles later if you're interested. One is with a company called Checkerspot. They produce specialized microbial oils that improve functionality of certain materials, right? So what we have done is we've been able to produce enough oil for them to generate the prototypes, and now Checkerspot has scaled up this technology to produce wonder skis. They're working with a brand to produce these specialized skis and many other materials right now. Another example, even more recent one, is a company which used to be called Joywell, now is called Oobli. They produce sweet proteins. Initially, we helped extract sweet proteins out of plant materials, and now we're looking at producing them microbially via yeast fermentations. So here you see ABPDU staff working with Oobli employees on the right. So one of the things that we have done, and we realized is very important, is co-location, or having our collaborators be on site when we're running their fermentation work or any kind of scale-up work for them, because our expertise along with their intuition really helps in minimizing any errors and salvaging any process that is going astray. So we do what we call affiliation process for our collaborators. So they become LBNL affiliates, do some safety training, and be on site. And this is a very important aspect for scale up, is that we really need to collaborate in person on site to be able to obtain successful scale up campaigns. And that's an issue when we don't have scale up capacity in the US and people have to travel abroad to be able to do the same. With that, I will thank the ABPDU team for doing all the wonderful work that I've just shown, and I'll hand over to Jim.
Thank you again, Deepti. And your slide there actually on downstream processing reminds me of a comment which I've remembered all the way through this thing about scale up from a colleague of mine from the chemical sector. And he said, in chemistry, the feedstocks are concentrated, the reactions are fast, and the products are concentrated. In bio, the feedstocks are dilute, the reactions are slow, and the products are dilute. And therefore, you spend a lot of time and money in downstream processing, which is just one of the many aspects of the problems of scale up in this business. So thank you again. Our next panelist is Dirk Carrez. And Dirk is the Executive Director of the Bioindustries Consortium, or the BIC, the BIC. BIC represents the private sector in a public-private partnership with the European Commission, the Circular Bio-Based Europe Joint Undertaking, or the CBEJU. BIC's mission is to connect industry, academia, regions, and citizens to transform bio-based feedstocks into novel, sustainable products and applications, and to create circular bioeconomy ecosystems through investments, innovation, and know-how. Now, that's a big remit. So Dirk is a bioengineer and holds a PhD in agricultural sciences. And Dirk, thank you for being with us, and over to you.
Thank you, Jim, and good evening, at least for me, to everybody. And I would like to thank the organizers to give me the opportunity to indeed explain a bit an initiative that we have set up in Europe to stimulate scale-up and deployment in general. And as Jim explained, this is a public-private partnership, which is at this moment called Circular Bio-Based Europe Joint Undertaking. Now, first, if this works, let me explain you what we are doing. So the Bio-Based Industries Consortium, or BIC as we say, is a consortium of around 240 companies, but coming from different industrial sectors, coming from agriculture, from food, from feed, forestry, pulp and paper, waste management, aquaculture, marine, chemical industry, materials, but also market actors. And this is very important, as you will see, because the focus is to set up value chains in Europe, from feedstock supply to new applications, and bringing also these products on the market. We have 4 main activities, 4 pillars. The first pillar is indeed being the private partner in this public-private partnership, the CBE, and I will explain that a bit later. Second pillar is what we call the business pillar. This is really making connections between different sectors, between companies of different sectors, because a side stream from a food industry can be a feedstock for a chemical industry. We also try to valorize all the components of a feedstock. And so you will have different products coming out for different applications, often different companies. So instead of having value chains, we're really setting up a kind of bio-based ecosystems, or biohubs, as we sometimes call it also. We also collaborate a lot with our associate members, which are the universities and research institutions. The third pillar is investment. How can we stimulate investments? How can we improve access to finance? Very important, certainly in Europe. How can we involve regions? And then the fourth pillar is what we call society. How can we increase awareness, knowledge, acceptance? And also the education pillar is very important. Now, the first pillar, as I said, is this public-private partnership. And this is a public partnership between our industry, the private partner, our members, and the European Union being the public partner. Between 2014 and 21, we had the first public-private partnership, which was called BBI, Bio-Based Industries Joint Initiative. And the European Commission provided, with approval of the member states in Europe, provided 1 billion euro to invest in research and innovation. And now the new Commission then, also last year, decided to give another €1 billion until 29 to invest in the follow-up, I would say, which is called now CBE Joint Undertaking. The private partners are investing several billions also, but they are doing that in their own projects, what we call in-kind participation, or additional investments, especially in the larger projects like demo projects or even flagship projects. What can be funded with this 1 billion public funding? Well, as I said, we try to bring partners of a value chain in projects. And that means from feedstock supply, the biomass supply, the biorefineries, the conversion, developing industrial applications, and even the brand owners bringing these new products and applications on the market. And this is very important. So we don't bring competitors together, or we don't give, I would say, grants to single companies, but always to a group of companies that develop a value chain. And there are 3 types of projects we can support. We have what we call research and innovation actions. That includes, by the way, activities and scaling up and pilot plants. And there we look to technologies that are still not mature enough to go to a demo plant. So how can we bring all these technologies to a level so that the whole value chain can go to a demo project? And a demo project are really proof of concept, small scale production plant, including developing new applications, so that at the end the risk for investment in a new production plant is lowered. And we can also give subsidies, grants to what we call flagship projects, which are really the first of a kind new innovative production plants in Europe. So this is quite unique. We can give support to these three kinds of activities, and of course the focus is really on upscaling, deployment, de-risking investments. What are the value chains? Well, we look to all kinds of products that can be produced, from chemicals, plastics, polymers, specialty chemicals, surfactants, cosmetics, textiles, and so on, from all kinds of biomass. And the biomass can be, of course, come from crops, can be side streams, can come from forestry, can be side streams from the pulp and paper industry, aquatic, algae, but also bio-waste, municipal solid waste, waste from food industry, and also CO2. So it's quite diverse. So what we really try to do is, with this research and innovation projects, we really, focus on, I would say, the fundamentals of, let's say, the future products, demonstration and innovation actions. We really, plant the seeds, I would say, for the future trees, which are the new biorefineries or flagship, first of a kind production plants. So we really go from research to, I would say, if these biorefineries, these first flagships, are replicated to the forest. We go from seeds to forest. That's really what we try to do with the whole program. If you look to what happened in the previous program, the BBI, which now ended, well, in green, you see that we have around 13 new flagships supported as projects within this BBI. And in orange, you can see all the demonstration projects all over Europe. And these demonstration projects are the embryos of the future flagships in Europe. And there, of course, the focus is also on upscaling, deployment. And on top of that, we have plenty of research actions, also including upscaling. And if you look to these flagships, this is very diverse. In the area of products. We go to building blocks, plastics, food and feed ingredients, proteins, fertilizers. So you can food and feed ingredients. So very diverse, but also the feedstock is very diverse. It goes from of course crops, but it also comes from side streams from, for instance, the sugar beet industry, microalgae, municipal solid waste. So also very, very diverse. What is nice, so we end up, it's really upscaling from research, and we end up not only with know-how, but we really end up with new production plants, new infrastructure, new investments. And these are some examples of new production plants that have been supported by these projects. For every, I would say, euro or dollar of grant, we have around 5.2 euros private investment. So the leverage effect is more than 5, very important, especially in these larger projects. And several of these flagship projects are now replicated in other parts of the world. So why is our industry participating in this kind of partnership, in this kind of projects? Well, as I said, first of all, it supports innovation. We try to go over through the valley of debt. So the focus is really on scaling up innovations to the size which is needed to become really a real investment, to be an alternative for fossil fuel. To de-risk investments, it helps if you have a proof of concept in a demo. It helps if you have a grant of 25 million also to find additional investors for a flagship. Of course, it are multidisciplinary partnerships. First of all, for the skills, but also, the different companies in the value chain that are needed. So the partnering with other industries, with also academia, are very important to develop this kind of value chains or ecosystems, as I said. Very important, we try to involve also brand owners. And of course, the whole idea is the focus is sustainability. And my last slide, one of the projects, by the way, of the BBI, you also have some funding for what we call support actions. And we have funded the support actions, which is called Pilots for You. You can find it on the website, which is a database mapping all existing open access pilot plans and the infrastructure that is available in Europe. And the reason we did this is that we didn't want to use public funding from Europe or from the member states to set up new pilot plants and to invest in new pilot plants, but we wanted to use that money, that funding, these grants, to support companies to go to existing pilot plants so that they could scale up their innovation. And this is a project that will go on now also under the CBE. So this is one of the examples that we try to develop in Europe to stimulate scaling up and deployment. Thank you.
And thank you, Dirk. It's I think our US colleagues will be interested to see that model. I've heard US colleagues in the past say they wish they had this type of model. For me and for you, Dirk, you'll remember before BBI-JIU got all started, the talk in Europe was always that we are great in the research, but we always miss out on demonstration. So to me, that's the most impressive part of this, because so many people forget that the demo phase, I would say, is actually essential before you go to full scale. And Far Eastern countries were never shy of building demo plants, but we always were. And therefore, I think that's a major achievement, is to get, I counted round about 60 demo plants into Europe and overcome that shyness of that type of investment. So thanks again for bringing that to us. And next up, I want to introduce you to the following speaker is Adam Lehman. Adam's a lead scientist of fermentation at the Good Food Institute with a focus on the development of biomass fermentation and precision fermentation biotechnology for food protein. Adam's technical background includes fungal genetics and systems biology, with industry experience in microbial, agricultural technology, human health studies, characterizing systems for strain development and process improvement. He holds a PhD from Drexel University College of Medicine in molecular biology. And then thank you. Over to you, Adam.
Thank you, Jim. And thanks again to the National Academies for sponsoring this. And this is really timely, and I think it's a really important topic. So as I move to my slides, I just want to introduce to you all the Good Food Institute. We are a 100% philanthropically funded think tank, and we operate to transform how people make and consume protein. So as we grow a population towards 10 billion to 2050, we're thinking a lot about reimagining food systems to make that food supply for the world more sustainable, secure, and just. And we do this in three main pillars through science and technology research, and that's where I sit. We research and fund and think about cultivated meat technologies, plant-based technologies, and then fermentation for food. We also have corporate engagement and policy efforts, and these are all designed to move towards our idea that we can reimagine this food system, not just here in the US, where we have the Good Food Institute, but through our affiliates as well across the world. So I wanted to start on a slide that has a bit of optimism. I would say that one of the things that we think about when we think about the challenges to scale up is that we haven't been here before, but in truth, we have. And often when we begin to biomanufacture, or I'll just use fermentation in a pretty wide definition, we're able to make that product very effectively. So you think back to the 1920s when people figured out how to make microbial produced citric acid very rapidly. With the technology of the 1920s, we were able to get almost complete market adoption of microbially produced citric acid compared to plant-based. When precision fermentation rennet, or chymosin B protein, was approved by the FDA in the US in 1990. It took only a few years to essentially take over about 90% of the entire rennet market. And these are good examples from food systems where you can see insulin is another great example. When we were able to take insulin and derive it from fermentation, we had almost complete market adoption as soon as we could make enough. And now here in 2020, we're sort of poised to do this in many different places at once. And we're thinking about now going from, particularly in proteins, making proteins that were maybe somewhat, I'm not going to say straightforward, but more simple to produce around 2020. And by the end of the decade, we imagine producing proteins that may be 5 times larger, more complicated folds. And we're constantly working towards higher concentrations and lower scarcity of these products. Now, fermentation and sort of biomanufacturing as an industry has been around for a long time, and we can think about 2 different sides of the pyramid, and this is a useful way of thinking about it. But if you go and think about pharmaceutical production and biologic drugs and things like that, these have been made at a high purity at a relatively low volume for a long time. And then if you think about things like biofuels, they've been made at an enormously high volume. Here in the States, that's really the #1 fermentation and biomanufactured product, but are made at scales and purities that may have a sort of different perspective than people who work in pharma. So where are we with everything else that we want to biomanufacture or ferment? We're somewhere in the middle. For food proteins, we obviously want to have high purity, but we want to have higher volumes than folks have in pharma. And we are going to be working with new technologies and new ways of producing these proteins that may borrow from relatively high value, low yield production systems. And we may borrow some technologies and adapt them from high volume fermentations and biomanufacturing. So this, I would say, from now until 2030 is an excellent opportunity to stake some of these foundational protocols and technologies in fermentation, but also in not just those bioreactors, but sort of that entire chain of what it takes to actually scale up a product. So again, we think mostly about food proteins, but I think you could take this end product here at a protein product and really substitute anything you want. And there are many challenges and many issues that we can deal with right now to deal with the scale up opportunities that we have. And here at GFI, we're taking a view that we can help to catalyze the change in many of these. And I can just briefly highlight some of the things that we're thinking about. So obviously, you need to feed whatever you're going to produce. So we're trying to perform, we are performing analyses to uncover unconventional or potentially new feedstocks that could go into our systems for fermentation. And these can be carbon beyond glucose. Obviously, we all want to use the glucose that we have to produce all kinds of biomanufactured products, and this could become limiting soon. Also nitrogen. There are challenges that are both geopolitical and technical with just getting nitrogen from our typical sources. So thinking about how we can get nitrogen, protein, and amino acids from ag and food side streams is something we actively think about. We think about workforce development, and that's not something going in your fermentation vessels, but they're the folks that are working at your facilities and the people that are performing the R&D that's necessary to make effective biomanufacturing systems. And our microbes are constantly getting better. And one of the things we do at GFI is think about how to plug in technical opportunities there. Currently, some of the scale up and challenges may come from, like we talked about, meeting people where they are and already using scale up facilities or piloting facilities, but there's also potentially how we could convert brownfields, and if brownfields conversion can be made economically feasible, then we have opportunities here to take some of the legacy capacity and property that we have for manufacturing in the US and worldwide and turn this into effective biomanufacturing facilities. Of course, we want to think more about geography and how this all fits into the economics of performing biomanufacturing. And if we can get facilities built or facilities retrofitted in places where the geography supports the workforce and supports feedstocks and distribution, then I think we're in the driver's seat on driving really this technology at scale and at cost. And again, this has been highlighted before, but processing technology is an enormous part of fermentation. We do have lower concentrations. We do have potentially smaller scales from each run. So we have to really be efficient. We have to valorize our waste and turn this into systems that can work for driving production that can be done at cost and that companies can continue to support, because it's not just the right thing to do, but then they can also continue to do it and keep the lights on. So I'll stop here and I'll give more time hopefully for some comments at the end. Thanks.
Yeah. Thank you, Adam. And I just want to touch on a really important aspect, which is often forgotten there, about brownfields. And I think you would be interested to talk to an Italian company called Novamont. And their practice in the past has to be buying old chemical plants that were defunct. Then they renovate the soil and they turn it into a bio-based plant. And whenever we've seen some sort of negativity in a community, they've saved the jobs and they've made jobs and they've renovated the soil, etc. The resistance disappears. So that's a company that has a lot of experience in just what you're talking about there, about geography and brownfield sites. So thanks for that. Yeah. And for our final speaker today is Alex Jaffe, is a product manager at Synonym. Synonym is a venture-backed startup working to develop, finance, and build commercial-scale bioindustrial manufacturing infrastructure. Synonym recently launched Capacitor, a free online database of microbial fermentation facilities worldwide. Alex holds an MBA from Northwestern University's Kellogg School of Management and a bachelor's in economics from the University of California at Berkeley. Prior to joining Synonym, she worked at Google for 7 years on several strategy and finance teams. So thank you, Alex, and over to you.
Thanks, Jim. Hey, everyone. Alex here from Synonym. Really glad to be here today. Today, I'm actually going to be doing a Capacitor product demo, which is a new free tool that we created to help users find and research fermentation production capacity. And I've actually created a video demo specifically for this seminar. So I'm just going to go ahead and play it now because I think that's the best way to show you all the tool. Today I'm going to show you how to use our new tool, Capacitor, which is the most comprehensive and free database of microbial fermentation infrastructure worldwide. Let's jump right in. To get the most out of the demo, I recommend you follow along on your computer. So open up your browser and go to www.capacitor.bio. Before we jump in though, I do want to share a bit more about why we created Capacitor for the greater SynBio community. Our goal is really to promote the growth of biomanufacturing by making it easier for SynBio companies to find the production resources they need to bring more bioproducts to market. We want to create more transparency and awareness around the global network of fermentation capacity and catalyze interest in building more. To accomplish these goals and bring the product to life, we teamed up with a few amazing knowledge partners. The Good Food Institute, a nonprofit think tank working to make the global food system better for the planet, people, and animals. Blue Horizon, a venture firm investing at the intersection of biology, agriculture, and technology to transform the global food system. And Material Innovation Initiative, a nonprofit devoted to accelerating the development of sustainable, animal-free, next-gen materials. Just wanted to give our knowledge partners a quick shout out for helping us bring Capacitor to our users. Now if we scroll down a little bit, we can see a brief snapshot of what's behind the curtains. As of now, we have 141 facilities listed in the tool. We can see that we have nearly 29 million of liters of capacity that's been uploaded. And lastly, we can see that there's facilities in 34 countries across the world. From the user side, we have users accessing the tool in over 60 countries, and that number just continues to grow. All right, now let's jump in and actually start finding facilities. We're going to click on the Get Access to Database green button, and if you're already signed in like me, you're going to go straight into the database. If you're not already authenticated, you're going to need to register or request a new magic link. Once you're in, you're going to see the map with the full view of facilities across the globe. On the left-hand side of the screen, you're going to see the filters that you can use to narrow down your search. On the right side, you're going to see a search bar and a preview of the facilities that meet your search and filter criteria. Now let's start actually filtering down the list. We're going to use the first filter, which is total liters at facility. Let's say that we're only looking for facilities that have between 10,000 and 50,000 liters of capacity. Let's type it in. You can see that the map changed and now only shows facilities that meet the specification. Now we're going to start going down the list of filters. For facility technologies, let's look at facilities that support fermentation. For feedstock, let's go ahead and select carbohydrates. It's important to note that if you use multiple filters, the results are going to use AND logic, meaning that both of these filters need to be satisfied in order for a facility to show in the results. For scale, let's go ahead and select pilot and demonstration. If you select multiple filter options within a single filter, like we just did, the results are going to use OR logic, meaning that either filter, pilot or demo, can be satisfied in order for the facility to show in our results. It's important to note that if you use multiple filters, for product expertise, let's go ahead and select proteins. And for size of bioreactors, let's go ahead and select 2,000 to 19,999 liters. I'm not going to go through the rest of the filters, but please feel free to try them out while doing your own searches. Now on the map, you can see the facilities that meet all of my criteria. On the right side of the screen, you're going to see the text preview of my results. Now let's say I want to look at these matches in more detail. I can either click on the marker on the map, or I can use the text preview to load the full facility card. Here we can see the entire detailed facility card for CPI Biotechnology, a facility that's located in the United Kingdom. As we scroll through the card, we can see the specs of the facility and get more information about things like downstream processing equipment. We can also see how recently the info was updated, and in this case, it was last updated 2 months ago, which is fairly recent. If we want to contact the facility, we can click on the Contact Facility button, and that's going to lead us to a contact page. Once you fill out the form, your request will be sent to the facility contact that we have on file, and then the facility can take it from there. There are a few other features that I want to show you. Let's say that we know the name of the facility or the name of the equipment that we'd like to search for. We can actually use the search bar on the right side of the screen to find this. I'm going to type in CPI, which is the facility that we just saw, and we can see that it pops up here in the results. If we click on the preview, I can view the full facility card and contact them as I just demonstrated in the previous map example. Now let's say I actually prefer to use the list view instead of the map view. Well, we can do that. If I click on the list button, it's going to lead me to a new view, which has the same functionality as the map view, except now I can search for criteria and I can search for countries. Let's say I wanted to search for facilities in India. I'm going to go ahead and type in India, and once I click on that, I can see all the facilities that are in this country. I can also use the filters on the left to narrow them down, and then once I click on a facility, I will see the full facility card as I showed previously. Now, let's say you actually have a facility that you'd like to list on Capacitor. Well, we made this a very easy process for you. If you click on the Add Entry form from the top of the page, you'll see a form to fill out. This form should only take a few minutes to complete, and once you submit it, our team will review it and publish your facility so that it's live on the platform. We really invite you to list your facilities on the tool so that this is the most comprehensive capacity database for users, and in turn, your facility will get lots of exposure to the hundreds of users who are registered and actively seeking production capacity. The last thing I'd like to show you is the FAQ page. As you're using the tool, we made a comprehensive FAQ page to help you get the most from the database. If you still have further questions, you can always reach out to our team at capacitor@synonym.bio or through the Contact Us part of the website. If you haven't already, we invite you to register and start using Capacitor today by visiting capacitor.bio. And if you have capacity, we'd like you to list it at capacitor.bio/facilities/new. Thanks for watching this demo, and we hope that you find Capacitor to be a very useful tool in finding the capacity you need to produce at scale.
So, Alex, are you done?
Yes, back to you, Jim.
Okay, so thanks for that, Alex. And I can say, well, that has to be whatever the other opinions would be, I'd say that just has to be useful to find those facilities wherever they are. Now, as usual, we're behind time. Always seems to be the case, and I want to open it up now to the panel for a couple of questions. I'll check in later with Andrew to see if we how many of these questions we want to actually address. But my first one for the panel is about technical factors. When you're building a new facility of any sort like this, you have many factors to build into the equation. The availability of a feedstock, regional facilities, different reactor scales, different process capabilities. How do you go about prioritizing these technical factors when you're setting out to make one of these facilities? And I guess, I mean, I don't expect everybody in the panel to go in a certain order. Some of you might even think that it's not the most appropriate for you, but I'll leave it to the panel to start to answer that question about how you prioritize all the different technical factors involved. Any takers? I know it's a difficult one. It's Dirk.
Well, I will try to answer. I think if I look a bit to Europe, because I know Europe, of course, a bit better. I think if I see in Europe, I think it all depends on the region. If you are in the northern of Europe, Scandinavia, what we call the bio-based industry is more forestry-based. You have some large research institutes like Luke, like VTT. And so many of these pilot plants are linked to these research institutes, and they have all the technical instruments that are needed, to upscale, especially forestry-based or related or forestry feedstock-related, let's say, technologies. If you are more in Belgium, France, Netherlands, that's more agricultural feedstock. So I think in our case, it really depends a bit on the feedstock available in the region, and then of course also to often if the pilot plant, for instance, is linked to a research institute, often there's a focus, and we have some, let's say, more independent pilot plants like the pilot plant in Ghent, Biobased Europe pilot plant, which is more an independent pilot plant that's more diverse. But I think it all depends indeed on the region, on the feedstock availability, and the focus of that region and the industries around it.
So, Dirk, I'm thinking you're what you'd call an expert in this area, but it strikes me if you wanted a non-expert system, some help would be available here from what Alex has just described, and maybe there's a case for a further extension of that type of facility. Alex, that's I'm suspecting that you have some of the answers within your database. It would certainly, what Dirk has said, it's take it from being expert down to non-expert.
Yeah, we have a lot of the technical capabilities listed in the database, so you can see the different specs that we are benchmarking the facilities on. So that can give you at least a good idea of maybe where to start and where to start prioritizing and ranking some of the different functionality that you can prioritize when you're looking at these very complicated facilities.
If anybody else has an input here, I've got a second question here, which I think actually it touches me in some of the non-technical parts, and I think they're equally interesting. And it struck me when Adam was talking that we come into some of these issues which are non-technical, and we've heard various people talk about workforce. And I had a conversation with Andrew the other day about what happens if you have the right feedstock and the right conditions down somewhere in the Dakotas, but the workforce that's available is mainly in California. How do you overcome that in a country the size of the United States? It's one of the worries we've talked through about this is you go through the process, and it's a long-term process of education systems for a workforce, and then what happens if the market's not ready and these people have to find jobs elsewhere? So are there any things in the panel that can tell us about how you acquire that well-trained workforce and at what sort of timescales we're talking about.
So I can chime in here real quick, Jim. So a couple of things. First, I want to respond to something you've asked earlier. You've asked how do we build biorefineries around feedstocks that are currently available. But I think we need to take a step back and think about how can we build technologies that will allow us to build biorefineries across, at least, let's say, the United States. And one of the things we looked at was blending feedstocks. So if you look at California, we have a lot of feedstocks, but there are a variety of them. And if a biorefinery is developed to be able to scale up only on a single feedstock, then yes, we cannot set up a biorefinery here. But if we can blend in multiple feedstocks and process them, and this is common in the petroleum industry, this does occur in the petroleum industry. So if we can do that, then we can start building biorefineries across the country, across the world. And that relieves some pressure around workforce development, like you've mentioned. So at the ABPDU, at least, we've had, over the past 11 years, we have over 100 alumni right now, many of them who are working at high-level positions in industry, some in academia even. And one of the things that we've seen is that people, after 2 to 3 years at the ABPDU, learn enough to be able to engage in industry problems or academic problems, whichever they choose. So it seems like we can train people for a period of time and put them at work, but we need 2 to 3 years of work experience for them to achieve that maturity, for them to go solve problems. So if we want to really develop a workforce, we need to engage now and build these programs that are a little bit more long-term. On the other side, on the flip side, what we have done at the ABPDU is we've generated a series of video SOPs for our equipment, standard operating protocols, because what we've also seen is sometimes we hire student interns. They come for one summer, they come back for the next summer, and they don't really remember a lot of what we've taught them. So we have to go through this retraining process a little bit. Having those video SOPs handy for them to actually just go and look at and come back, so they're not bothering a senior engineer, taking time away from actual work, is very, very helpful. And also, we want to be able to use it to engage with number of community colleges and just like really scale this process of training as fast as we can through virtual tools, and then bring them on site for a short period of time. So yeah, on one side, we need to really scale fast virtual tools for education. And on the other side, we need 2 to 3 years timelines on training to get that type of quality workforce that we need to solve real problems.
Well, I'm glad you mentioned the word community colleges, Deepti, because it's one of the things I've talked about quite a bit. If you talk to the academic community about skills and education, you get very quickly to the PhD level, but that doesn't build a workforce. If you fill a factory with PhDs, you'll get a million ideas and probably no products. And one of the things that's European countries have been, well, some are better than others at apprenticeship programs. And one of the aspects of apprenticeships is what we used to call the dairy lease, where an apprentice would work full-time in a company and 1 day per week would go to a college, not to a university, to learn some of the more academic aspects of the business. So there are many ways to look at this. And because, since what we're talking about California, it's my understanding that the largest educational system in the world isn't the University of California, but the community colleges system of California is the largest in the world. And my understanding also is that many community colleges are often strapped for cash. So you could kill more than one bird with one stone here, with a single stone here. So, Andrew, I'm wondering, is it time to get out to the audience? Because we've, mostly because of my big mouth, we're running a little bit behind time.
Yep, we can add them, I guess, to call. I saw that you had unmuted before Jim sent you the message.
Yeah, okay. I can be pretty brief here. Jim, I think you hit the nail on the head with community colleges. There's programs. So here I'm based in North Carolina that are training people at the community college level for plant operators and biotech facility operators and things like that. And I think that's perfect. And we need to get them into the general biomanufacturing field as well, not just pharma. And on that note too, I think there's a lot of leveraging that we can do from other industries and meeting people where they are. If you look at bioprocessing, you can look at some of the ideas that they may be able to get and the training that they may be able to get from agricultural processing, from dairy processing, people who work in oil crush facilities or people that work in dairy protein facilities really could have a lot of the same skills that would be highly overlapping. So I think there are a lot of opportunities. We just have to go out there and find out and identify those opportunities for people entering the workforce or looking for a job.
Yeah, and I think a final part I would just add to that, Adam, is this really actually takes strain away from universities. Universities do research, are great at research, and these kind of skills are probably not the kind of things they want to learn. So the community colleges benefit, and the universities benefit by able to concentrate on what they do best, essentially. Now, I have a question for Dirk here, and it's, do you have suggestions of public-private partnership models where incumbents make their proprietary technology available to startups without losing their competitive advantage, and thus everybody benefits? Have you any thoughts on that?
First of all, I have to say that there are no specific rules or obligations in the projects of this public-private partnership. So the grant is given to a consortium, and it is a consortium that decides in their consortium agreement how the IP will be, for instance, also developed and treated, who will be the owner, what is already known before the project, and so on. Also, I have to say, and maybe if you talk about public-private partnerships, many people think this is about also large companies. 80% of our companies, our members, are SMEs, what we call SMEs. So small and medium-sized companies, or even micro companies, so startups, or a bit more mature than a startup. And 40% of the funding, of the total funding of all these grants, are going to these small companies. We have many of these demo projects where the leader is an SME, where the majority of the funding goes to a small and medium-sized company. And this is crucial because that's the only way for this company, to go to a demo, to go to a proof of concept, and together with other companies. And two, in this consortium, you don't have the competitors. You have additional companies that, one for the feedstock supply, one is developing applications. So you don't have competitors, but you really need partnerships. You have to create synergies. So up to today, we never had any problem about this I would say IP issues or these technologies. And many of these by the way, small companies are also technology providers. So they have a platform technology. And the large companies are very happy that they can use that for a specific product. And then, of course, the discussion is, of course, who will have the rights on the product. But that's part of the consortium agreement between the partners in a consortium.
Well, you mentioned during our presentation that you're looking for complementarity when these partnerships are being built. So quite clearly, you don't want people who are in competition to do the same thing. You want people with different companies with different skills to make your value chain, yeah. Now, I have a question here for Alex, I guess. It's, is it possible to use the information gathered by Capacitor to help identify the biggest needs and gaps for building scale-up facilities in the US? Is there a better tool for understanding the needs in the US?
Yeah, I think we definitely can use Capacitor to help understand that issue. I think what we can do is look at things like the total liters and also the product expertise and technologies that the facilities support, and then pair that with some of the demand numbers that we're seeing to see what is the demand for certain technology capacity, certain product expertise capacity, to figure out what do we have, what do we not have, what do we need to build, and where do we need to build it. Because we have inventoried a lot of the capacity that exists in the United States. We're not fully done yet, but hopefully we'll get there. And so we can really see what that underlying data looks like in the tool, and then, hopefully get some demand numbers, whether they come from GFI or elsewhere, and see what are those gaps and where do we need to build. So yeah, that's definitely something that we can get, and we are looking into getting from Capacitor.
Right. But it's almost invariably when we do exercises like this, you come through at the end and say, oh, we're out of time, and you desperately wonder how you can then keep the conversations going. But for now, unfortunately, we've reached the limit of our time, and I'll great discussion. Many of the things that we know are the problems are addressed here, and if you'll notice, the 4 different speakers there were from different areas with quite different experiences, and let's hope we can maybe learn something in the future that can keep these kind of discussions going. But I'll hand you back to Andrew now for the final words. So thanks for your time, and thanks for your patience.
Thank you, Jim. And I might actually use my privilege to ask hopefully a 10-second question to Adam. I'm curious, and Alex too, if you follow on, if a similar tool exists for brownfield facilities, and if that sort of kind of an inventory of potential facilities that could be refurbished is needed. And Alex, I guess, if Synonym is looking to kind of build that inventory as well. But maybe Adam first, is there an inventory that exists that's helpful?
There's not an inventory that exists. So I would say right now we're still building out some of our knowledge here about what's possible with those brownfields, and then what the numbers look like on those conversions, and then see if there's an opportunity. But I do think that, given the amount of brownfields that exist in the U.S. and worldwide, if we can come up with a sustainable way of converting them, we can create the demand to address and find all of those.
I guess you need to be talking to the EPA.
And I would echo what Adam said. It's definitely been something that's been on our radar. It's not part of the current functionality of the tool, but since it's a very important aspect of this particular ecosystem, it's something that we're looking into in the future.
Great, thank you all, and thank you for those last couple of minutes over. So, and thank you, Jim, for moderating. I want to just kind of close out. Thank you to all of our panelists for all of your contributions to the webinar today. And as I mentioned at the start, this is really just the first of several follow-on events to our October workshop. If, as I mentioned as well, if you have any input or insight on recommendations on future topics, or would like to stay informed and are not already on our mailing list, please do reach out to my email, which is located on the website. Thank you all again for tuning in, and wishing you all a wonderful holiday season and an early New Year's. Thanks.
Thank you.